Hi everyone, my name is Kipp, and I'm a PhD student in the veterinary medicine program at the University of Minnesota College of Veterinary Medicine. I'm excited to talk to you today about my ongoing research that applies some cutting-edge, miniaturized, and mobile molecular tools for the surveillance and study of tick-borne pathogens. The diseases spread by ticks have an enormous public health impact, representing approximately 95% of all locally acquired vector-borne disease cases in the United States. Although ticks have long been known to serve as disease vectors, novel tick-borne pathogens continue to be identified in recent years. The epidemiology and collective public health impact of these new and emerging tick-borne diseases is still poorly understood. One particular tick species, the black-legged tick, Ixodes scapularis, has a disproportionately large impact on public health. Although many people are likely familiar with Lyme disease, caused by the bacteria Borrelia burgdorferi, the general public is largely unaware of the many other bacterial, viral, and parasitic diseases that Ixodes scapularis is capable of transmitting. The necessity of tick surveillance is underscored by the fact that over the past 2 decades, the total number of tick-borne disease cases reported in the United States has nearly doubled as vector populations have expanded and human-tick contact continues to increase. Molecular surveillance of these pathogens is an essential public health tool. However, most conventional surveillance approaches are hindered by requiring large laboratory footprints and an extensive time between sample collection and screening results. There is currently a significant need for improved approaches to tick-borne pathogen surveillance, particularly in ways that facilitate timely and comprehensive screening results for multiple tick-borne pathogens. Recent technological advances have fostered the development of miniaturized and portable devices that have formerly only been available to large, well-established laboratories. Our group has developed a mobile lab composed of many of these newly developed miniaturized molecular devices that is capable of rapidly conducting screening assays to detect both known and potentially unknown tick-borne pathogens. The mobile lab is highly portable, can be run largely off battery power, and takes up about as much space as a standard suitcase. This allows all components of the mobile lab to be deployed across a variety of remote field settings. Using the mobile lab, tick collected in the field can be immediately processed for nucleic acid extraction. Then, a multiplex, probe-based real-time PCR approach using a small, portable thermocycler can simultaneously screen for 3 pathogen targets and provide surveillance results from time of sample collection in a matter of hours. In parallel, the same extracted tick nucleic acids can be subjected to nanopore sequencing, the next-generation sequencing approach that has a wide variety of metagenomic applications. In addition to providing confirmation of our PCR screening results, nanopore sequencing data from the mobile lab can be leveraged to characterize patterns in the tick microbiome and can facilitate pathogen discovery efforts to identify pathogens for which we lack a priori knowledge. We have recently had the opportunity to demonstrate the feasibility of our mobile lab through screening a sample of 52 adult Ixodes scapularis ticks collected across 2 field sites in Minnesota. By screening ticks individually, we were able to successfully demonstrate that we could detect the pathogens Borrelia burgdorferi, Anaplasma phagocytophilum, and Babesia microti. Our mobile lab also identified a single tick with the recently recognized disease-causing agent, Ehrlichia murus occlurensis. Interestingly, we found that 25% of all ticks collected screened positive for 2 or more pathogen targets, and we are actively investigating tick co-infection patterns and the role that tick co-infection may play in disease maintenance. Collectively, these preliminary results support the utility of the mobile lab in the surveillance and study of tick-borne pathogens across a variety of remote field settings. As we move forward with this research, our ongoing efforts will continue to explore the efficacy of nanopore sequencing for characterizing metagenomic samples from ticks. We are actively planning on deploying the mobile lab to further demonstrate its feasibility and its flexibility across field settings in the coming year. I would quickly like to thank all the co-authors of this infographic and all the individuals who have assisted on this project thus far. Thank you for taking the time to listen.